Let me start KGRI seminar. So my name is Hideki Okano, Department of Physiology, University School of Medicine. Okay, so it's my great pleasure that I can introduce Dr. Akira Yoshi from Developmental Anatomy and Cell Biology, Pediatrics and Neurology at University of Illinois at Chicago. Actually, I have known him more than 25 years or something like that, and after he did undergraduate training in Department of Physiology in Keio University, and so his current position is assistant professor at Department of Anatomy and Cell Biology and Pediatric Neurology in University of the Illinois at Chicago. And so in 1995, he graduated Keio University School of Medicine and got an MD. And so when he did a residential training at Department of Pediatrics at Keio University Hospital, and then Pediatric Neurology Service at the Massachusetts General Hospital. And after that, he worked in for 4 years training in as MGH, and then he did a research scientist in developmental neurobiology at MIT at Professor Martha Constanti's Patton's laboratories. And he did he's published his PhD work, and it's in Nature Neuroscience. Actually, I have been one of the committee members for his PhD defense, and it was very excellent and outstanding paper. I was very impressed. Okay, and then he got a position, and the current position at the University of Illinois. He's working in both clinics and basic of the pediatric neurology, and he's going to make a talk today. The characterization role of Palmitoyl-protein Thioesterase 1 in developmental plasticity and CLN1 disease. Okay, Yoshi Sensei, please.
Well, first of all, I'd like to thank Dr. Okano and the audience here today for giving me opportunity to give my recent work. So in my lab, we are focusing on the activity-dependent synapse formation in both health and disease. And this is the generic synapse, excitatory synapse in electron microscope. And as you can see, axonal terminal that is filled with a bunch of synaptic vesicles, and that is opposed to postsynaptic membrane. And this is the tip of dendritic spine, and which is stemming from dendritic shaft. And as you can see, this dense membrane called postsynaptic density, or PSD, that has covers both receptors and associated molecule, and receive neurotransmitter signal and convey the signal to postsynaptic neuron. And the postsynaptic density, or PSD, is not a simple structure, and according to several mass spec study or proteomic study, there are about thousands of different proteins. And this is just representative proteins, including neurotransmitter receptors like glutamate receptor, including NMDA receptor and AMPA receptors. And these molecules are held with a scaffolding molecule called PSE95, and that is also bound to the second layer and the third layer of postsynaptic density molecules like a GCAP and SHANK, which is associated with the autism spectrum disorder or Phelan-McDermid syndrome. Anyway, the point I'd like to make on this slide is many of these surface molecules are modified by a small lipid called palmitic acid, which is a component of palm oil, basically. This palmitic acid is very important protein modification because it allows these postsynaptic PSD molecules for trafficking from the cell body to the postsynaptic membrane and helps clustering and make help form postsynaptic density. And the other thing I'd like to mention, which is important for this talk, is the regulation of glutamate receptors. And here we have NMDA receptor, which is the major glutamate receptor. This receptor goes through developmental subunit switch. And here you have the we have the neonatal form or very immature form of composition of NMDA receptor, which has NR2 subunit or GLU-N2B subunit. And the NMDA receptor is a calcium passing ion channel and held by scaffolding molecule SAPON2. At some point during development, this composition of the receptor is switched to NR2B containing receptor or GLU-N2A containing receptor. And this is also the calcium passing ion channel, but the amount of the calcium is much lower than the neonatal form. And this is very important for the maturation of dendritic spine. And even in adult, the NR2B and 2A receptors are both important. And the immature synapse, the NR2A or GLU-N2A receptor subunit containing receptor is GLU-2A subunit containing receptor is distributed mainly at the postsynaptic membrane, while 2B containing receptor is pushed away from the synapse and reside in extrasynaptic area. And the difference between these two receptors is first, it has a different response to synaptic plasticity, but also it has a different response to the something called excitotoxicity, or when the neuron, the presynaptic neuron releases overload of glutamate. The postsynaptic neuron undergoes the cell death, and this process, excitotoxicity, is mainly go through with NR2B containing receptor. And here, there's the second type of receptor, AMPA receptor, and this is the much mobile than NMDA receptor. And when NMDA receptor is sensitized by glutamate, the calcium enters, and there's a whole host of the response in signaling cascade, including chem kinase, and changes phosphorylation status of AMPA receptor. And depending on high frequency or low frequency stimuli of the presynaptic neuron, there are two forms of synaptic plasticity known as long-term potentiation or depression, LTP or LTD. And when LTP occurs, more AMPA receptors are inserted to synaptic membrane, and in low frequency stimuli, when the neurons are not is stimulated with low frequency stimuli, the AMPA receptor is retracted or to the intracellular spaces, and this is recycled. And and in this slide, I'd like to point out the one of the principal postsynaptic molecule, PSE95. And in my previous work, I've shown that PSE95 goes through several steps of trafficking mechanisms in order for the molecule go from the cell body to the postsynaptic membrane. And first, it depends on the neurotrophic molecule like a BDNF, brain-derived neurotrophic factor, and which stimulate track B signaling pathway. And depending on the PKC, the molecule, the palmitoyl-protein enzyme, and there are about more than 20 different enzymes that belongs to ZDHHC, and in this case, ZDHHC8 allows the PSE95 to be palmislated. And once the PSE95 is palmislated, it sticks to the transport vesicle, and this vesicle, the transport is activated by again from BDNF from track B, but the intracellular pathway is different. PI3 kinase and AKT goes through or facilitate the trafficking of PSE95 containing vesicle. And once this vesicle is transported from the dendritic shaft, it switches a molecular motor and the cytoskeleton within within dendritic spine, which is enriched with actin, and the PSE95 uses molecular motor called myosin 5A, and eventually go to postsynaptic density or postsynaptic membrane. And so we have a good idea about how synaptic molecules are transported from cell body to postsynaptic membrane. But there is a turnover or proteostasis of these and other synaptic PSD molecules, as well as presynaptic molecule too. And so there's a very little understanding of how the retraction of this molecule or turnover of this molecule occurs And first of all, you could imagine that these molecule undergoes endocytosis, and depending on the activity, it may go back to the postsynaptic membrane, but eventually all molecules become old, or there may be an overload of these proteins. So eventually, these molecules has to be digested, and there are a few pathways to digest these molecules. The first is the proteasome complex, and the second is lysosome, and this is the easy way to understand the function of the lysosome is the garbage disposal of a cell, and there are many different enzymes within lysosome, which is acidic environment, and the before these proteins, especially the proteins that are modified either sugar chain or lipid, has to go through the enzymatic retrieval of these modifications, whether it's a sugar chain or lipid. And in this case, the enzyme that is responsible for, and we think that is responsible for the cleavage of palmitic acid, is a protein called PPT1, or palmitoyl-protein thioesterase 1. And this molecule is quite important because it is associated with a rare, or I should say ultra rare disease, and lysosomal stress disorder called neuronal lipofuscinosis, or NCL. And this disease has a long history, and it's been known since the beginning of the last century. As such, there's many physicians who describe the similar findings and symptoms, and it has a lot of different names. But the more recently, because of the molecular genetics, it was reclassified, and it is still called NCL, or Batten disease. But the most recent nomenclature depends on the responsible gene, which are CLN1 through 13. And so at least there are 13 different phenotypes that correlate with genotypes. And many of the protein product of these CLN genes are either lysosomal enzymes or the molecule that is important for the biosynthesis of these molecules. And we are focusing on CLN1, or PPT, and the gene itself, the protein itself, is the palmitoyl protein palmitoyl thioesterase 1, or PPT. And so this is the most aggressive form, and that happens in starting from babies, and it is also called infantile neuronal lipofuscinosis, and in the or it is also called CLN1 disease. And in this disease and any and other CLN1 and disease have shared clinical symptoms and neuropathological findings. And first of all, they have sort of like in case of infantile form, it started with psychomotor regression around 6 months. They don't start to sit up. They're still bedbound, and they don't even roll over. And then they start to have some sort of visual symptoms and blindness and retinal degeneration, but also it had the disease affect the central visual cortex too. And then they start to have seizures, and in juvenile onset or adult onset form, they start to have daily confusional states, and it also almost mimics schizophrenia. And then they start to have respiratory symptoms, and eventually they die. And in case of infantile form, the average lifespan is about 2 or 3 years. So this is a devastating disease, but it is very rare. It happens 1 in 100,000. So, but I'd like to pay attention, draw your attention in the neuropathological finding. And here you can see this inclusion body called the lipofuscin or autofluorescence lipopigment, because without any fluorescence, you can actually see these material on light microscope. And this lipofuscin is not specific to NCL in general, but it is also present in adult onset disease like Alzheimer's disease, the Parkinson disease, and ALS or motor neuron disease. So we think this is a common denominator of the neurodegeneration. And the final stage of this neurodegeneration is cortical atrophy, as you can see here. And by understanding the mechanism of the CLN1 and other CLN, we'll be able to understand the basic principles of how palmitoylation is degraded through this protein networks that is associated with CLN genes. And hopefully we can come up with a therapy or a therapeutic target and that can translate into adult onset disease. So to understand the functions of these PPT1, we started forming a hypothesis, a very simple hypothesis, in which the lack of loss of functional PPT1 result in the lack of depalmitoylation, and the overloaded molecule start to form the inclusion bodies that is seen as autofluorescent lipopigment, but also there are overloads of functional proteins in both pre and post-synaptic receptor membranes. And because of the overload, it can start to have all kinds of bad things, like starting from seizures to excitotoxicity and so on. So with this simple hypothesis, we first looked at the neurons from PPT1 knockout mice, and in the in today's talk, the part of the PPT1 story is single-handedly performed by a very talented graduate student, Kevin Costa, who is now a postdoc at the University of Chicago with Murray Sherman's lab. And so he started observing the enlarged lysosome, which is stained with LAMP2 that co-localize with autofluorescent lipopigment, and it is larger and it is engorged as compared to the wild type neurons. And he used the palmitoylation inhibitor so that there are fewer palmitoylated molecules, and as he predicted, the accumulation of autofluorescent lipopigment is much less as compared to the without treatment. And he used two different palmitoylation inhibitor, and this is a quantification of the size of the inclusion body, and which is reduced. The size is reduced by the two different palmitoylation inhibitors. And next, he looked at the morphology of dendritic spines, and the top panel is wild type, and the bottom is the mutant, and the knockout neurons have a thinner and longer dendritic spine, or almost looks like a phylopodium, which suggests that the PPT1 knockout neurons are the immature neurons as compared to wild type neurons. To look at the biochemical correlation, he used immunoblotting and looked at the ratio of GLUN to A and to B. And so remember the 2B, the higher amount of 2B is more immature, and more 2A means more mature. And as predicted, the ratio of 2B and 2A shows a higher amount or a higher ratio of 2B as compared to 2A, indicating that the biochemical component of the postsynaptic membrane is immature. And this correlates with anatomical structure of the thin and long phylopodium looking appearance of the dendritic spines. And next, with the help of electrophysiologists Walter and Julia, they looked at the NMDA current at P40 postnatal day 42, and this is a timing that is way beyond the developmental switch of NR2B to 2A. So at this point, you predict to have the mature component of NR2A, and the way we looked at the subunit components are the measuring the tau, or the half-life of the curve, and the shorter tau means there are fewer calcium passing, and the wider tau means more calcium entry. indicating that there's more to be contained in the receptors. And at this age, you think that the NMD recurrent is very narrow, so that there's more to A-containing receptors. And as you can see, the comparing wild type and the knockout, the PPT1 knockout mutant mouse has wider tau, suggesting that there's more NR2B component, which is the immature NMD receptor component. And then we also looked at the calcium imaging in culture cell, and this is the wild type, and the calcium is shown with is visualized by genetically encoded calcium sensor called GCAMP. And let me just turn off the pointer. And this is how the calcium increase look like. And the higher intensity of the calcium signal is mainly localized within dendritic spine, and this gives the impression that it almost looks like a Christmas tree. So that's the wild type. The mutant, the PPT1 knockout neuron shows quite different pattern, and instead of the punctate increase of the calcium is mainly increased in dendritic shaft, and it's wide range of calcium increase, and which is so like the signature of the NR2B containing receptor. And when the Kevin treated neurons with palmitoyl inhibitor, instead of now the calcium is compartmentalized within dendritic spine, and much fewer, less amount of the increase in dendritic shaft. So this is the quantification, and as you can see again, the PPT1 knockout, there's a wider range of increase in calcium, and the lower change with the palmitoyl inhibitor treatment. And so next, he looked at the response to the excitotoxicity, and this is the cell death induced by hyperactivity of neuron, and that depends on mainly NR2B containing receptor. And eventually, whether it's wild type or knockout neurons, they all die. But during the first 20, as the composition of the chemicals that induce NMD receptor dependent excitotoxicity, there is depending on the ratio, at some point the PPT1 neuron shows a higher death cell death rate, and so this argue for the increased susceptibility to excitotoxicity in PPT1 knockout neurons. So finally, he Kevin looked at the palmitoylation state of NMDA receptor subunit and 2B and 2A. So this is a method called APEGS assay to specifically look at quantifying palmitoylation of a molecule that you are interested in. When you read this Western blotting, we take a ratio between non-palmitoylated band versus palmitoylated band, and so not just intensity change of the all bands. And when he took the ratio of each molecule, each cultures, he found that the glutamine-2B subunit, which is the immature form of NMDA receptor subunit, is increased as compared to wild type, which is in line with the previous data. And this is the palmitoylation is suppressed by the treatment of the palmitoylation inhibitors. And this didn't happen in N2A mature form of any NMDA receptor subunits. And so then we are not really sure of how to interpret these findings, and we scratch our head and went on to screen for several postsynaptic molecules, and it turned out a molecule called FIN, which is a non-receptor tyrosine kinase, and is associated with Alzheimer's disease. Its palmitoylation state is also increased, and the role of FIN in the context of the NMDA receptor subunit switch is it keeps 2B, which is the immature form of NMDA receptor subunit, on the membrane. So this thing tries to maintain the immature state of the synaptic development. And so this is the model we configured, and early on, there is a trafficking of the glutamine-2B containing receptor, and FIN try to retain the 2B containing receptor at the synaptic membrane. And at this stage, there is a very little expression of PPT1, and that happens in the wild type or normal development. And when the PPT1 start to kick in, there is a protein turnover, so that the amount of the FIN at the synaptic membrane becomes a little lower. And because it's lower, the 2B start to be dispersed around the extrasynaptic membrane. And while the mature NMDA receptor containing 2A subunit start to be inserted. But in the disease state, without functional PPT1, the FIN start to accumulate at the synaptic membrane, so that the more 2B containing receptor is retained at the postsynaptic membrane, and this causes the retention or the retention of the immature NMDA receptor, and disrupt the maturation of the synapse. Okay, so this is the already published in eLife a few years ago, and now we switch a gear to the second receptor, AMPA receptors. And by way of introduction, the AMPA receptor is important for at least two different forms of synaptic plasticity. And one is the LTP and LTD that we already discussed. And in aggregate, LTP or something like LTP or LTD that depends on the rule of fire together, wire together, is termed Hebbian plasticity. And the second form of plasticity are called homeostatic plasticity or synaptic scaling. And this form of plasticity is sort of like compensatory mechanisms. And and this is not just in single neurons, but the networks, a whole network of activity. And when the activity of the circuit increase, the postsynaptic neurons have a compensatory response, and it reduces amount of AMPA receptors so that it balances the activity of the local circuit. On the other hand, if there is any sort of sensory deprivation that causes lower network activity, something like dark rearing or monocular deprivation, then there is also a compensatory mechanisms in postsynaptic neurons by increasing the amount of AMPA receptors, so that with lower activity, there is the more sensitive postsynaptic neuronal activities. And so in both mechanisms, AMPA receptors plays a critical role. And the other thing I should mention is in Hebbian plasticity, the response is immediate. Whether it's a high frequency or low frequency stimuli, the increase or decrease of the AMPA receptor or the electrical signal happens immediately after the synaptic stimuli, while in homeostatic plasticity or synaptic scaling, this is a compensatory mechanism. So it takes a few days to readjust the amount of the AMPA receptors. So in case of the monocular deprivation plasticity, it takes about 2 to 3 days in order for homeostatic plasticity to occur. With that, the synaptic scaling, as I said, depends on the surface expression of glutamate receptors. And normally, in case of the LTP or LTD, the composition of the glutamate AMPA receptor subunit is equal ratio of GLA1 subunit and GLA2 subunit. But in case of the homeostatic synaptic scaling, the composition of the inserted AMPA receptor depends on the AMPA receptor that is exclusively configured with GLA1 subunit, as you can see in this figure. And when this in this composition, the AMPA receptor in normal composition of the AMPA receptor, the calcium doesn't pass, but in the GLA1 containing receptor is calcium permeable receptors, and And this can be confirmed by a specific inhibitor called NASPAM. And with a and there's the paper that the scaling when it is scaling is induced, upscaling is induced by tetrodotoxin for 48 hours treatment. There's more AMPA receptor current, but is the NASPAM treatment, which blocks GLUA1 containing receptor, blocks the synaptic scaling or upscaling. So, which is shown by the lower AMPA receptor current as compared to without NASPAM treatment. And there's a new study looking at the palmitoylation proteomics and looking at the effect of the PPT1 knockout. And in this study, they compared wild type and PPT1 knockout mice and looked at the amount of palmitoylated proteins or synaptic proteins. And sure enough, they found the GLUA1 subunits. So this encouraged our study to look into synaptic scaling. And as you can see, in this graph, we induced synaptic downscaling by treatment of AMPA receptor inhibitor by tubuling. In control, there is a reduction or downscaling of the AMPA receptor in Western blotting, but that doesn't happen, indicating there is an occlusion of downscaling. And next, we looked at upscaling by suppressing all synaptic activity by treatment of TTX for 48 hours. And then there is a increased surface expression of GLUA1 subunit, which is shown in many investigators. And when we looked at the same induction of the upscaling in PPT1 knockout, there is an increase of the GLUA1 subunit, but it's exaggerated, much higher amount of GLUA1 subunit, indicating that there's a hypersensitivity or overreaction of synaptic upscaling. And in the next several studies, we try to confirm the upscaling phenomenon in different ways. And the first way is to not just biochemically, but anatomically or immunohistochemically look at the distribution of GLUA1 containing AMPA receptor on the cell surface. This is a staining without permeabilization method, so that we can only see the surface expression of AMPA receptors. Sure enough, we looked at the upscaling of GLUA1 subunit in wild type, but as indicated in the Western blotting finding, we also saw the exaggerated response of the expression of GLUA1 subunit in the upscaling induced by 48 hours treatment of TTX. And so this is the another way of looking at the activity of GLUA1 containing receptor. And remember, I said the GLUA1 only receptor is calcium permeable receptor. So we took advantage of that and looked at the calcium increase within a cell using, again, GCAMP6, which is the genetically encoded calcium indicator. And they quantified the increase of the calcium shown in this signal. And the undergraduate Ibrahim, basically collected all synaptic dendritic spines that has increased at some point of imaging, and blindedly Kevin analyzed the calcium increase and looked at the response to NASPAM in 2 different concentrations. And this is the lower concentration of NASPAM, which is the GLUA1 containing receptor inhibitor. And in both wild type and the knockout, the NASPAM reduces the calcium entry, but there is a higher decrease, or much well, higher, looks like a lower, but it's actually higher amount of the calcium block with low concentration. And so then in higher concentration of NASPAM, we saw the same trend, and there is a higher sensitivity to the NASPAM inhibition in the PPT1 knockout mice, but we also saw the dose-dependent decrease in calcium. So this is encouraging because it's not just responding to inhibitor, but higher higher dose of inhibition result in higher decrease in calcium, calcium entry. So then next, we decided to look at the synaptic scaling in vivo by using dark rearing. So this is a very common way to decrease the sensory activity. And the previous studies show that the dark rearing induces a higher amount of anti-receptor that depends on the GLUA1 containing receptors. And so we looked at the amount of GLUA1 first in control normal rearing, a light rearing environment. And we noticed the reduction of the GLUA1 subunits, but with the dark rearing in both wild type and in knockout, there is an increase in GLUA1 expression. And when we analyzed the ratio between the dark rearing and light rearing and the dark rearing in each genotype, although wild type also showed the upscaling, the percentage of the upscaling, the increase from here to here is higher in the knockout mouse. Okay, so we now have a good idea that the PPT1 knockout mouse have the higher sensitivity or higher induction of homeostatic upscaling, but with the increased GLUA1 containing AMPA receptor, what happens next? And what molecular pathway GLUA1 containing receptor activates? So that's the next question we wanted to address. And with the help of the biochemist Stephanie Colonia and Ming Guan in the chemistry department, we did the palmitoylation specific protein in awake animal. Sorry about the technical glitch, and I just wanted to show in the last 1 minute the 2-photon imaging. And in wild type, you see the sporadic the activity of GCAM signal in the local circuit, and that happens in here as well. But you can see that there's at some point there's a wave of the network, the hyper synchronized activity, and with the quantification of that such signal, we are able to visualize the higher synchrony of network activity in excitatory neurons as compared to wild type. And this is the statistics, and again shows the higher activity. And there is a computational study that higher homeostatic activity leads to the hyperexcitability, leading to something like epilepsy by Sinofsky's work. And we are quite satisfied with what we found. So with that, I will close and just thank the member of my lab where is the acknowledgement? Okay, I can't find the acknowledgement. Okay, here. Okay, great. Okay, so I'd like to answer any question that either in person or